Okay, are there any questions? I'm not hearing anything, so I'll assume there aren't any questions. Let me turn on the sound. So you should be seeing my screen. We're going to cover Chapter 8, Microbial Genetics, or I should say continue Chapter 8, Microbial Genetics. Remember, you have a quiz this Friday. Quiz 2, covering Chapters 4, 5, 6, Labs 2, and 3. If you haven't taken the practice quiz 2, it was actually due yesterday, but you can take it today. You will get 2 extra credit points there, as well as getting practice for quiz 2. And it uses the, what is it called? The lockdown browser. So if you take the practice quiz, you'll be in good shape for taking the quiz 2. All right, any questions about what we're going to cover? We probably will finish Chapter 8 today, in which case I'll do a little bit of review for the quiz too. Don't know what that is. So I believe this was the last slide we had talked about yesterday. Can anybody confirm that?
We were starting at operons.
We're going to start on operons shortly. There's one other slide, I did briefly mention it, and that is in eukaryotes. The RNA is transcribed in the nucleus, and that doesn't happen in prokaryotes, but in eukaryotes, the RNA is transcribed in the nucleus, and the genes of eukaryotes have introns in them, so that there are regions of the gene that are transcribed as messenger RNA which do not code for protein. Or if we're talking about a tRNA or an RNA, the introns do not code for the tRNA or the RNA. And those introns in the RNA transcript have to be removed. And that processing happens in eukaryotes. In fact, an RNA transcript made in a eukaryote will not leave the nucleus. So these introns in the RNA transcript prevent the RNA from leaving the nucleus. It's not until the introns are removed that the final RNA transcript, which we'll call a messenger RNA, leaves the nucleus to go out and be translated as a protein. And what happens is that the introns are spliced out, that means cut out, and then the exons that code for the functional protein, they're spliced together, meaning they're put together. And then you have the final messenger RNA, which now can leave the nucleus. And so that is RNA processing in eukaryotes. Any question about that? So now we're going to switch on to the regulation of bacterial gene expression, and we'll talk about operons and how they regulate gene expression. We're done talking about the structure and function of the genetic material. And then we'll move on to mutation, and then the genetic transfer and recombination, and lastly we'll end with genes and evolution. So bacteria want to regulate their contents, and an excellent way for a cell to regulate its contents is to regulate the level of gene expression. So if you don't want a protein in a cell, you can simply turn off the gene. And if you want the gene, then you turn on the gene. So that is one way to regulate the contents of a cell, by controlling gene expression. There are other ways to regulate the contents in a cell. You can have have enzymes active, or you can inactivate enzymes. Any question about that? We're going to talk about regulating the expression of genes. There are some genes that are always turned on, and so they're always expressed at a fixed rate. And these we call constitutive genes, such as a constitutive enzyme. The cell always makes these enzymes, so the gene is always turned on, and the gene is expressed at a fixed rate. Any questions about that? There are other genes which are expressed only when they're needed, such as repressible and inducible enzymes. When the cell doesn't want this enzyme around, it turns the gene off. And when it wants the gene around, the enzyme, it will turn on the gene. And there's two ways to do that, meaning two ways to regulate the pre-transcriptional control, meaning you can turn off or on the gene, and there's inducible operons, which those genes are normally turned off, but they're turned on when the cell wants them. And then there's repressible operons, which we're not going to cover, and you're not going to be tested on it. But when in a repressible operon, the gene is normally turned on, and it is turned off when the cell doesn't want it. There are other ways to control the gene expression. There's epigenetic control mechanisms, and I mentioned there's post-transcriptional control, where you can control the protein after it's made, such as inactivating an enzyme or activating it if it's inactive. We're not going to talk about any of those. So we talked about genes, and they can be expressed, turned on, or they can be turned off. Does it make sense for a cell to make a product if it's not needed? Would it make sense to a cell to make this product if it's not needed? Can anyone answer?
No, it's not. It doesn't make sense to make it if it doesn't need it.
Yeah, if it doesn't need it, it doesn't make sense to make it. So the cheapest way to regulate that is just to turn off the expression of that gene. Okay? And that's what cells do. If they don't need something, they do turn it off. Now, I'm going to talk about this as simply turning the gene off or on. Please understand that it's much more complex to that. You can have a gene turned off, but you can have it turned on at very low levels. You can have it turned off at low levels. You can have it, I guess that's turned on at low levels. You can have it turned on at moderate levels, and then you can have it turned on at very high levels. And we're not going to talk about the complexity of that. We're only going to be talking about genes being turned on or turned off. Any question about that? It's kind of to simplify things. So how a gene controls the level of gene expression is by using operons. And we're going to talk about the operon model of gene expression. A bacterial operon is sort of like the super gene. An operon contains the structural gene or genes that we're talking about. So we'll be talking about the lactase gene. Let's see if I can scroll that down or blow this up. So we're going to be talking about the lactase gene. It is on the lactose operon. The lactase gene is the Z structural gene, and that is the actual DNA sequence that codes for the enzyme lactase. Any questions about that? Now, in the lactose operon, let me get my mouse working, you have the lactase gene, but you also have the gene that codes for the transport protein to bring the sugar lactose into the cell membrane. And that's the Y gene. And then the lactose operon, or the lac operon as it's called, also codes for a third structural gene, and this is the gene that helps convert beta-galactose into the sugar glucose. So this gene switches lactose into beta-galactose and glucose. This gene brings the sugar lactose into the cell, across the cell membrane in a transport protein. And then this gene helps convert beta-galactose into the sugar glucose. And all of those are in a bacterial genome. They are on the lac operon. The lac operon encodes, besides the structural genes, it also encodes regions around the structural genes that help control the expression of the structural genes. And we'll talk about these. They're controlling regions of DNA, and that includes the promoter, which we've talked about, and the operator, and we'll talk about that. And these are controlling regions around the gene of interest that help express or turn on or off the structural gene. And we're specifically talking about the structural gene lactase. And then there's another thing that isn't shown on here on the operon, and that's the terminator. I think we talked about that in RNA transcription. It follows the last gene, and it codes for the RNA polymerase to fall off the gene, the DNA, and then that ends transcription. And so the terminator is also a part of the operon. All right? So those are all of the parts for the operon. And you can think of an operon as a super gene, meaning, yes, it includes the gene lactase, which is normally what we think of when we talk about genes, but it also includes the controlling regions that determine whether the lactase gene is going to be turned on or off, the terminator for ending transcription. And in bacteria, you can have other structural genes on the same operon. Any questions about that? Now, the lac operon is an inducible operon, and that means that it is normally turned off, but it can be turned on when the cell wants it to be turned on. Okay? Any question about that? Now, the regulatory gene is a separate gene, not on the lac operon, and it helps regulate the expression of the lac operon. Normally, the regulatory gene is nowhere near the gene or genes that it controls, meaning the lac operon. But in the case of the lac operon, the regulatory gene is right next door to the lac operon. In a eukaryote, the regulatory genes that control the expression of the operon usually are on a different chromosome, so they're nowhere near the operon. But in the lac operon, the regulatory gene just happens to be right next to the lac operon. Now, we are talking about a operon in bacteria. It even says right there, bacterial operon. When we're talking about operons in eukaryotes, they are the same. They have a promoter, they have an operator, they have a terminator. How the eukaryote operons differ is they only have one structural gene for each operon. And so eukaryotes have the same genes, a gene that codes for lactase, a structural gene that codes for the enzyme lactase, and a protein to bring in the sugar lactose across the cell membrane, as well as the gene to help convert beta-galactose into glucose. But in the eukaryotes, they have them on 3 operons. In bacteria, they simplify it and they put all of the related genes together on the same operon. So in a way, eukaryote operons are simpler. There's only one structural gene in an operon. However, eukaryotes are more complex in their operon, and that is they have many more regulatory units controlling the expression of an operon. Any question about any of that? All right, so we mentioned that in bacteria, such as E. coli, the lac operon is normally turned off, and it's inducible, which means that it can be turned on. It can be turned on. Why do you think in E. coli the enzyme lactase, or the lac operon, is normally turned off? What do you know about the sugar lactose that E. coli would normally turn off the lactase enzyme?
Does it have to do with the carbon structure of the sugar?
No, something even simpler than that. How often do you find the sugar lactose? Where?
Just in milk, I think.
Yeah, just in dairy products. Sorry, for some reason this is having an echo. At least occasionally. You only find the sugar lactose in dairy products. So unless you're E. coli on a dairy farm, you're not going to be around the sugar lactose. And so if you're not going to be around the sugar lactose, because lactose is just not very common, there's no reason to turn on the gene. So in E. coli, the lac operon is normally turned off because E. coli doesn't usually see the sugar lactose. Any question about that? Well, hopefully you understand that because E. coli doesn't normally see the sugar lactose, it normally turns off the lac operon. Right? You know, I can't see anybody, so I don't know if you guys are getting this. Everybody's got a black picture except for one lady who has, two ladies who have a picture on there. So I'm not seeing anybody's expression, so I'm hoping you're getting this, because I can't tell. There we go. Thank you, Christopher. So, how is the lac operon turned off? Because that's normally the way the lac operon is in E. coli. Well, what happens is the regulatory gene, which is the I gene, is transcribed, and it has its own operon that we're not going to talk about, okay? And when the messenger RNA is made, it makes the repressor protein. And then the repressor protein binds to the operator of the lac operon. Now when the RNA polymerase binds at the promoter region, remember the promoter is part of the lac operon, and it codes for the RNA polymerase to bind to the DNA here. And then that promoter moves down the lac operon, transcribing messenger RNA as it's going. Well, when the promoter moves down to the operator and the repressor protein is bound to it, the RNA polymerase can't move any further, and it's stopped. And so it never transcribes the lactase gene or any of the other genes of the lac operon. I'm talking about structural genes. And that shuts down the lac operon. Any questions about that? It's actually a very simple method to shut down the lac operon. The repressor protein is active and binds to the operator, and then the RNA polymerase can't move down the lac operon. What happens to that RNA polymerase? Do you think it's stuck there forever?
It gets released back into the new
yeah, it's
cytoplasm,
released
doesn't it?
back into the cytoplasm, because we're talking about a bacteria here. That RNA polymerase isn't stuck there forever. It's binding with a stoichiomic reaction, and so every once in a while it falls off, and that will free it up for binding to another promoter. Perhaps the same promoter, but another promoter. Meaning that RNA polymerase isn't stuck there for eternity because it's stopped by the repressor protein. It'll eventually fall off, and then it can go bind to another promoter. All right? So that explains how the lac operon is turned off. Let's explain how it is induced so that the expression of the lac operon occurs, and then we get the lactase gene made, as well as the transport protein to bring the sugar lactose across the cell membrane, and then the gene to help make beta-galactose into the sugar glucose. And they do have different names. Generally, beta-galactosidase, that's the same thing as lactase, and since that's an easier name to say, that's the word I usually use to describe this enzyme. Permease is the protein, that's the active transport protein that brings the sugar lactose across the cell membrane, and I don't even want to talk about that one. All right, so what happens to turn on the lac operon is that there is an inducer which essentially turns on the lac operon. And in the case of the lac operon, the inducer is allolactose. So I think lactose, I don't remember, is a left-handed stereoisomer, if you remember what a isomer is. And then the same molecule, but it is right-handed, is allolactose. So it has the same, what do you call it, carbons and oxygens and all that. So it's the same sugar, allolactose. The only difference is it's a mirror image of the sugar lactose. Everyone clear on that? It's an isomer of lactose. So whenever you have the sugar lactose around, you have allolactose around, because for some reason, if you only had one, over time, the sugar converts, don't ask me how, I don't remember, from one to the other. And then you'll have both of them around. But normally in the environment, you'd have both around anyways. Anyways, allolactose is the inducer, and it will bind to the repressor protein. And when it binds to the repressor protein, it inactivates the repressor protein. Now the repressor protein cannot bind to the operator, and so there's nothing bound to the operator, and if the RNA polymerase binds to the promoter, it now transcribes the lac operon, making the structural genes of interest. Okay? Any questions about that? There should be one. What happens to the repressor protein that's bound to the operator? Come on, let's have somebody else answer besides Christopher. What happens? It's bound to the operator. How do we remove that structural protein? Okay, Angeline, do you have a guess? What happens to the repressor protein in this situation when allolactose is around? Angeline's not going to guess. How about Sierra? Do you want to guess? Come on, Sierra, don't be shy. All right, Sierra's not guessing. How about Alejandra? Alejandra, do you have a guess as to what happens to the repressor protein? JC, do you want to guess? Leona, Ian.
Does the protein fall off into the cytoplasm?
Yes, that protein is bound by a stoichiomic reaction. We ran into that once before, if you remember, in the operon. And every once in a while it falls off. If there's other active repressor proteins around, one of them will bind to the operator. But in this case, when the repressor protein falls off, there's the inducer around and it can bind to it, and then that will inactivate it, and then there's no active repressor protein around to bind to the operator. So any questions about that? So we're not always stuck in this situation where that's bound to it, because every once in a while it falls off, and if allolactose is around, it'll bind to it and inactivate it.
What happens to the inactive protein?
I'm sorry?
What happens to the inactive protein in the cytoplasm?
Oh, what happens to the inactive protein? It doesn't bind to the operator. However, every once in a while, the allolactose will fall off of the complex here, and so that will make active protein. And if there isn't allolactose around, it won't bind to the active protein, so it'll bind to the operator. So the question is, I've never had a student ask that. That's actually a very good question. What happens to this? And that is when allolactose becomes rare in the cell, it starts falling off the repressor protein, and then there's not enough allolactose around to bind to the repressor protein, so it'll bind to the operator. All right, any questions about that? So that is the way that E. coli and bacteria control the expression of the structural genes in the lac operon. When the sugar lactose isn't around, then allolactose isn't around, and that means the repressor protein is active. It binds to the operator, and that shuts down the lac operon. Whenever the sugar lactose is around, allolactose is around, it will bind to the repressor protein, and so if it's bound to the operator, it'll fall off and then become inactive. And then the RNA polymerase can bind to the promoter, move past the operator, and then transcribe the structural genes, in which case we get the genes expressed in the lac operon. Now, what do you notice about this messenger RNA, which is a little unusual, because we probably never discussed this? How often have you seen a messenger RNA that codes for more than one protein?
Those specific to prokaryotic?
Yeah, this is specific to prokaryotes. This only happens in a prokaryotic operon where there's more than one structural gene in the operon. So in a eukaryote, you'd never see this. A eukaryote would only have one protein made from a messenger RNA. In prokaryotes, it depends on the operon. But if there's more than one structural gene in the operon, as is the case for the lac operon, you can get more than one protein from one messenger RNA.
Would the lac operon being turned on be considered an environmental factor?
Yes, the environmental factor that turns on the lac operon is whether allolactose is around in the environment. If allolactose and lactose aren't around, the lac operon will be turned off. But if lactose and allolactose are around, the lac operon will be turned on. Good question. All right, any other questions about operons in general?
Can operons cause different growths in bacteria?
Do they cause different growths? Well, if there's no other sugar other than lactose around, if the cell has the lac operon, then this bacteria can grow. But if the cell
different
doesn't
morphology.
I'm sorry?
Different morphology.
I don't understand what you're asking.
Just different traits in the same species of bacteria could be caused by operons being turned on or off?
Yes, different traits in bacteria can be caused by operons. Like in the case of the enzyme lactase, it is not made unless the sugar lactose is around. Okay? So different traits can be turned on depending on the environment that the bacteria is in. All right. Let's move on to the topic of mutation. A mutation is a genetic change in the DNA. Mutations may be neutral, meaning they have no benefit or harm to the individual, like this eye color probably is neutral. They may be beneficial, or they may be harmful. If you want to know, most mutations that happen at random are actually harmful. The second most common would be neutral ones, and we'll talk about them. And very rarely is a mutation beneficial. I think we talked about the blue-eye gene being a mutation that happened in the human population around 10,000 years ago. That gene nowadays is neutral, that it does not give an individual an advantage. But they think 10,000 years ago that that genetic mutation gave a selective advantage to those who had it. And that's why that gene increased in number in Northern Europe. In fact, blue-eyed gene, the blue-eyed allele, is the predominant gene in Northern Europe. And that's where the mutation happened. So they think a long time ago, like 10,000 years ago, that that mutation was beneficial. All right? And whatever that benefit was, it no longer exists today. A mutagen is an agent that causes a mutation. You could say increases the rate of a mutation. And that could be something like X-rays increase the rate of mutation, or I think it's nicotine, but I could be wrong. I don't remember. I should remember this, but I don't. I actually studied that when I was in graduate school. I think nicotine in cigarette smoke is a mutagen. Benzene is another mutagen. Okay, a chemical agent that's a mutagen. Mutations can happen spontaneously, and we call that spontaneous mutations. They occur in the absence of a mutagen, and they occur all the time in the human genome. It's just that they're very rare when they happen. Generally, when a gene is being copied, you have to copy about 1 million genes before you get a mutation. Okay? Any question about that? Now, that's very rare, but when you say that the population in the world is, what is it, 7 to 8 billion, something like that? Do I have my names right, my numbers right? Yeah. So 1 billion, and you only need 1 million to get a spontaneous mutation. It tells you in the human genome, we're getting spontaneous mutations all the time. Any question about that? There are different types of mutations, and this is actually in the worksheet, but I'll probably briefly go through it. You can have a base substitution, that's a point mutation, where one nucleotide base is switched. And that's why it's called a point mutation. There are different types of base substitution. You can have a silent mutation, which you'll learn about in the worksheet, a missense mutation, and a nonsense mutation. A nonsense mutation codes for a stop codon. A missense is where you change the nucleotide base and it codes for another codon, meaning another amino acid. A silent mutation is where you change the nucleotide, the DNA, but the protein is not changed because the codon codes for the same amino acid as the original codon. Is that clear? I can show you the codons if you need to see that. Like if we had AUU and we switch it to AUG, don't ask me if those code for the same amino acid, but they might. And if they did, that would be a silent mutation where the DNA is changed, but the protein is not changed because the codons code for the same amino acid. And silent mutations are always neutral mutations. Any question about that? I thought I heard somebody talking, so. All right. You can also have a frame shift mutation. A frame shift mutation is when one or more nucleotides are removed from the DNA or are added to the DNA. I should really show you an example of a codon to talk about that. Let me bring up a new thing here. So if we have a G, T, C, A, G, G, T, T, C, C, A, oops, sorry, not a capital. And we should probably put those in codons so that they're easier to follow. If you have a frame shift, let's go A, and then we remove the G, that now throws off the codon. Do you see this? So frame shift mutations, when we remove the G, that changes the amino acid from this point all the way down because it now reads the codon differently. Instead of being CAG, we remove the G, so that C comes up to the first codon, and then the next codon is AGG. And all the other codons are changed. So a frame shift mutation really changes the protein. Does everybody see that? And it would be the same thing if I added a nucleotide. That still would be a frame shift mutation. You change the codons from where you added the nucleotide.
Yeah.
Okay? Any questions about that? All right. So that's a frame shift mutation, and it really does change the protein after where the frame shift mutation is. Now let me state that you could pull out 3 nucleotide bases or insert 3 nucleotide bases. And that just pulls out, let's talk about pulling out, that just pulls out 1 amino acid from the protein. And then there's no frame shift after that. So that's actually not a frame shift mutation. I'm not sure what that mutation is called when you pull out a group of 3 or you insert a group of 3. You pull out a codon or you add a codon if it's a group of 3. And so that's really not a frame shift mutation. Okay? And then there's a mutation which is not discussed in the book, and I have no idea why the textbook is not addressing it. It's a macro mutation, where an entire region of a chromosome is either added or deleted. or you could have, in rare cases, an entire chromosome is added or deleted. Okay? And that's a macromutation, where you're changing not just one gene, you're changing a large number of genes. And you actually know an example of that. Let me scroll that up. Can anyone tell me a macromutation that's quite common in the human population?
Trisomy 21.
Yeah, Down syndrome, trisomy 21. I think that's right. Where an entire chromosome is extra in those individuals. Okay, there's another one, there's actually a couple of them, and that is there are females born with only one X chromosome. That's not a lethal mutation, although they're not reproductive. And then there's individuals who are born with two X chromosomes and a Y chromosome. So those are macromutations. Okay? And they're common ones in the human population. Normally, when an individual has an extra chromosome or is missing a chromosome, the embryo will die, meaning you can't muck up the genome that greatly. But in Down syndrome, whatever chromosome that is, I think it's 21, it might be 23, I don't remember. When you add an extra chromosome, it's not lethal for that individual, but it does have an effect. They have too many genes that are expressed, and that results in Down syndrome. Any question about that? Anyways, that's a macromutation. And like I said, you have to learn that from the lesson because your book will not discuss it. Let me see what I want to talk about with this. You can have a base substitution where the wrong base is inserted and it doesn't pair well, and because of that the cell could actually repair that, but we're not going to talk about DNA repair. What happens is if the wrong base is inserted, you get a spontaneous mutation, and the G will be okay, and so that line will be correct, okay? But this line, the wrong nucleotide will be inserted next to the T, so that'll be an AT, and then we'll get, when the DNA replicates, when this DNA replicates over to here, we'll then get the mutation if we follow this line of DNA. Any questions about that? Oh, here I've got showing you a silent mutation. I didn't remember I had this slide here. And let me see if I can blow that up a little bit. If you change the base from UUU to UUC, that's a silent mutation because both codons code for phenylalanine. So there will be no change in the protein. A base substitution is when you change the nucleotide. Instead of being what the heck's the codon here? GGC maybe? Yeah, GGC, and it's changed to AGC. This is a missense because you code another amino acid here. And so there will be one amino acid change in the protein. Was somebody asking a question? I'm not going to talk about examples. A nonsense mutation is when you change one base, and instead of coding for a codon that codes for amino acid, you now code for a stop codon. And what this will do is that instead of making the complete protein, you will make a truncated protein. Any question about that? Here's the frame shift mutation, which I've already talked about, so let's move on. All right, so we talked about mutations. Mutations can be spontaneous, where they're simply made when DNA is replicating and a mistake is made in the replication of DNA. And that's a spontaneous mutation. Mutations can also result from a mutagen. And mutagens can either be physical, like radiation, or chemical, like a chemical mutagen, like benzene would be a chemical mutagen. There are two types of radiation that can cause mutation. There's ionizing radiation, such as X-rays and gamma rays, and when they hit an atom in a molecule, like a carbohydrate or DNA, they cause the formation of ions. Usually what they do is they give so much energy to an electron in the atom that they collide with, that electron is shed from the atom, and then that makes an ion. And then this ion can react with DNA and cause a mutation. That is how ionizing radiation causes mutations. It can create an ion directly in DNA as well, but most of the time it's creating an ion in the cell, and then that ion bumps into the DNA and mutates the DNA. Another way that radiation can cause mutation is by non-ionizing radiation, and that is UV light or UV radiation. When UV light strikes DNA, it creates a covalent bond between two thymines. And that's hard to see here, but the two thymines pair with each other covalently, besides at their nucleotide, meaning in the sugar phosphate backbone, the two bases are actually binding with each other from UV light. And they do not bind with the A's. And so that makes a bubble in the DNA because these thymines are binding to each other, not hydrogen bonding with the A's. All right? And that can mutate DNA because actually that thymine dimer will stop RNA transcription and DNA replication, and if DNA replication occurs on the thymine dimers, it will not read it correctly, creating the mutation. Chemical mutagens such as nitrous acid can act as a mutagen, and what nitrous acid does is it binds to a nucleotide such as the A here, and then it chemically modifies this A. It is still adenine, but it's slightly different. And now this A does not pair with T. This mutated A will pair with C, and then when this goes through DNA replication, that C will create the mutation. Instead of being CGA, will be CGG. Any question about that? So that's how nitrous acid acts as a mutagen. How are we doing on time-wise? Oh, we still have a little bit of time. As I mentioned, Spontaneous mutation happens all the time. It actually happens when 10 to the 9th bases are replicated by DNA polymerase. One of those bases will be a mistake and will not be caught and will give rise to a mutation. Now, it is true that the cell could perhaps catch that mistake and then correct it, but we're not going to talk about DNA repair. Okay, so we're going to say that spontaneously mutation happens at about 1 base in 10 to the 9th replicated bases. Now, a gene on average has about 1,000 bases, so that's why we say that one gene in a million, when they're replicated, will be mutated. Any question about any of that? Mutations increase the rate of a spontaneous mutation. Mutagens generally increase the mutation rate by 10 times or 1,000 times. And that means they increase the rate or probability of a mutation from 1 in 10 to the 6th genes being mutated to 1 in 1,000 or 1 in 10 to the 5th replicated genes being mutated. All right? Any question about any of that? Now, how can we identify a mutant? It can be done experimentally to test for mutation, and there's 2 ways to do that, meaning 2 ways to test for if there is a mutation. The first is a positive or direct selection. You detect the mutant cells because they grow or appear differently than the non-mutant cells. Like if anybody saw this person in the population, you'd say, yep, that's a mutant. Okay? Any question about that? There's also negative and indirect selection, and that is when you're detecting the mutant cells because they do not grow. And that's probably not terribly clear, so let's discuss negative indirect selection a little further. I did this. Let me blow that up a little bit. You can make replicas of colonies that you have on a plate. So here you have colonies that have been plated out, dispersed, and you can make a replica of this plate by getting a velvet pad and then pressing it onto the plate, and then taking this velvet pad and placing it on 2 other plates. Well, if you use the pad to place it on one plate containing media that has the amino acid histidine in it, and another plate that is lacking the amino acid histidine, and then you look at the growth on these 2 plates, you can see that this colony here and here, that in the media lacking the amino acid histidine, this colony is not growing, but it is growing if we supply the amino acid. So we can say that this colony here is a mutant, and that is it will only grow if the amino acid histidine is supplied in the media. And that is a negative selection for a mutation. Any question about any of that? All right. So that's it for mutation. Is there any questions about mutation? All right, let's move on to genetic recombination then. Genetic recombination is the exchange of genes between two DNA molecules. And that can happen when crossing over occurs. When you have crossing over, two chromosomes get together, and one could be from the parent, and all of the alleles are lowercase. So let's say that chromosome's from the father, and this chromosome is from the mother, and the mother's chromosome has all of the alleles uppercase. The two chromosomes can come together and then cross over, and then they break in the crossover, and then this region of that chromosome is added to this chromosome, and that region of this chromosome is added to this chromosome, so that we reassort the genes on the two chromosomes. Now we have lowercase e, lowercase f, lowercase g, but uppercase H and uppercase I, reassorting the genes. That's all genetic recombination does, is reassort the genes so that they can be assorted differently, meaning the lowercase g is now assorting with the uppercase H. Okay? So recombination, all it does is it mixes the genes around in the population so that they can be expressed in new combinations. Any question about that? No questions? Then I'll ask you a question. Are recombination and crossing over the same thing or something different? Does anyone want to take a guess?
They're different.
It obviously depends on how you're looking at it. So if you're being very strict about how you're looking at it, they are different. But then if you're not strict on how you're looking at it, they're sort of the same thing. They both simply recombine the genes so that new expressions of genes can be expressed. But yes, they are different in the sense that recombination is just talking about genes, and then the genes recombining, whereas crossing over is specifically talking about the chromosomes recombining and breaking.
Isn't crossing over much more similar to meiosis?
Sorry, can you say that again?
Isn't crossing over much more similar to like meiosis?
Crossing over in humans generally only happens in meiosis. That's when the chromosomes recombine or get together, and then they can cross over. Okay? So generally in humans, that's when crossing over happens. And generally in human, that's when recombination also happens. Okay? But recombination specifically talks about the genes recombining, and it's a little more general than crossing over, because crossing over is talking about the chromosomes coming together, pairing, and then breaking, and then reassorting. Okay? So it depends how you're looking at it, if they're the same or if they are different. Okay? Because generally, the same thing happens. Whenever you have crossing over happen, you have recombination happening. And generally, when you have recombination happening, it happened because of a crossover event. All right? But like I said, recombination is a little more general of a term than crossing over. All right. So let's talk about genetic transfer. We talked about vertical gene transfer, and that occurs during reproduction between generations of cells. So one parent cell can split into two daughter cells. That's vertical gene transfer. And in humans, it's two parents. The gene transfers vertically into an offspring. Okay? That's vertical gene transfer. Well, there's also in cells horizontal gene transfer, where the transfer of genes happens from one cell to another, but the two cells are in the same generation. So DNA leaves one cell, that's the donor cell, and then it enters another cell, that's the recipient cell. And horizontal gene transfer can happen in three different ways. It can happen by transformation, conjugation, and transduction. Any question about any of that?
Is the sex pili a type of horizontal gene transfer?
Yes, we're going to talk about that when we talk about conjugation. So maybe I should talk about that right now. Conjugation happens in gram-negative cells because of the sex pilus. Conjugation can also happen in gram-positive cells, and it happens in the mating bridge. I'm not really going to talk much about mating bridges, but when gram-positive cells get together, they can actually fuse their cytoplasm, and then they have a channel where the cytoplasm from one cell can move into the other cell, or vice versa. And they can exchange genetic information through this mating bridge. All right? We're not going to talk further about gram-positive cells, but we will talk about gram-negative cells because they can engage in conjugation through sex pilus. The sex pilus is this long structure coming out of an F+ cell, and it has the sex pilus, and the sex pilus will literally skewer an F- cell. And then that sex pilus will winch the two cells closer together. But they don't have to become right next to each other because the sex pilus is actually hollow. And that will allow a plasmid to move from the F+ cell, which is called the F plasmid, and into the F- cell. So that's how the sex pilus performs conjugation. Okay? And this is showing you what happens. The F plasmid is in the F+ cell, and the F plasmid has the genes on it to code for the sex pilus. So an F+ cell is the cell that has a sex pilus. Any question about that? And when the sex pilus skewers an F- cell, it can bring them together, but they don't have to be this close together because the sex pilus is hollow. That F plasmid will then replicate and send a copy into the F- cell, and that's happening here. So that both cells now have an F plasmid. Okay? And that will convert this F- cell into an F+ cell because this cell now has the F plasmid, which codes for the sex pilus. So this cell will form a sex pilus. And then this cell can go out and skewer an F- cell and engage in conjugation with the F- cell. So that's why this cell is converted from an F- cell to an F plus cell. Any questions about sexual conjugation? Let me state that this cell gains new genes that it did not previously have. And that is, it never had any of the genes on the F plasmid, and now it has all of the genes on the F plasmid. So it gains new genes, and it expresses a new phenotype, and the new phenotype is the sex pilus. Any questions there? All right, that is conjugation. Let's talk about transformation. Transformation, make sure I'm on the right slide, is where DNA comes out of one cell and then enters a recipient cell. And in transformation, the DNA is naked DNA that enters the recipient cell. So the donor cell that this DNA came from, in nature, the donor cell would die because the cell does not lose its DNA. Any question about that? And in a lab, you can have transformation happening all the time, and people take DNA out of the donor cell, so the donor cell is dead, and then they combine this DNA with the recipient cell to get recombination or transformation. Okay? Anyways, the recipient cell takes up the naked DNA, and then that naked DNA, part of it or all of it, can recombine with the chromosome, and then this cell can get new genes that this cell did not have. Like this cell had the capital A gene before. I'm trying to make my mouse work. And from the DNA we have the lowercase a, and after the transformation, this cell now has the lowercase a. That can give this cell new phenotype. Like suppose lowercase a codes for antibiotic resistance. This cell now will be resistant to that antibiotic. Any question about that? How important is transformation as well as conjugation and transduction? Well, mutation happens in about 1 in 10 to the 6 genes. So if we're looking at the gene for antibiotic resistance, it happens in bacteria only 1 time in 10 to the 6 cells. Okay? By spontaneous mutation. By transformation in bacteria, it happens 1 times 10 to the 5 cells, meaning it happens 10 times more frequently. Any question about that? With conjugation, it also happens about 10 times more frequently, in gram-negative cells at least, than in spontaneous mutation, or 1 in 10 to the 5th cells. And the same thing with transduction. It happens 10 times more frequently than mutation. So these other ways of getting horizontal gene transfer are important to cells because they can get a new gene by one of these 3 ways: transformation, conjugation, and transduction. And each of them are happening 10 times more frequently than are happening by spontaneous mutation. All right, I have a cute little picture of transformation. A cell sees a piece of DNA and brings it inside, gets some indigestion. The genes are expressed from this DNA, and then the cell is transformed. Any questions? All right, let's move on to transduction. Transduction
Oh, Dr.
happens...
Wolpe, It's 2:16.
Oh, 2:15. Yeah, all right. Let's go ahead and stop right here, and I'll come back at 2:30. Any questions?
Would carcinogens be considered a type of mutagen?
Carcinogens typically are mutagens. There are a few exceptions, meaning the correlation is not 100%, but generally carcinogens are mutagens, meaning most of them.
Okay, thank you.
Okay. All right. I'll see you at 2:31 probably.